Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we were discussing about cell biology, in which we have covered the part about cytoskeleton, and then we started discussing about plasma membrane. So we have already discussed about the models that were given to explain the structure of plasma membrane. Then we learned about the composition of plasma membrane, the structure of plasma membrane in detail, and then we also learned about the properties that plasma membrane possess and on what factors those depend. So now in today's class we'll be learning about the important characteristics of the lipid bilayer, the asymmetry of membrane. So so you must have seen the thumbnail. I've drawn a schematic of the plasma membrane where you can see it is only simply lipid bilayer. It doesn't have protein, so it is protein-free lipid bilayer. And the lipid bilayer that I have drawn, it's quite symmetric. But on the contrary, in the cells, the membrane, the lipid bilayer is asymmetric. Means the composition of the both of monolayers, the composition of the leaflets, it is different. So if the lipid bilayer was symmetric, this means the different types of phospholipids that are there or the sphingolipids that are there, the percentage of them that will be equal in both of the leaflets. But this is not the case in the plasma membrane. For example, the phosphatidylcholine that is present in higher concentration in the exoplasmic monolayer. On the contrary, phosphatidylserine, inositol, and phosphatidylethanolamine, they are present in more numbers on the cytoplasmic monolayer. And that is what creates the asymmetry of membrane. So the lipid bilayer is asymmetric. Oh, well, asymmetric means the composition of both monolayers is different. So let's say we plot a graph representing the percentage of lipids that is present in lipid bilayer. So on the y-axis, I'm going to plot the percentage of the lipids present. Okay, this is percentage of lipid molecules. And on the y-axis, I'll be plotting different types of lipids. So this is a bar plot. So let's list out the different types of lipids that are found in the membrane. So in the membrane, we find phospholipids. Okay, we find glycolipids as well as we find sterols. So we can see cholesterol. Okay, I'm representing it as Cl. Then we can also find phosphatidylcholine, we can find phosphatidylserine, we also have phosphatidylinositol, phosphatidylethanolamine, and the sphingolipids such as sphingomyelin. Okay, so all of those, they are present in different concentrations, in different proportions in the lipid monolayers. So the sphingomyelin, if we start plotting the percentage of lipid molecules sphingomyelin, that would show a graph like this. So the graph above the zero, okay, on the x-axis is actually shows the exoplasmic monolayer, while the below panel, the below leaflet, is the cytoplasmic monolayer. Then the phosphatidylcholine would have a graph something like this. The phosphatidylserine, something like this. This is ethanolamine. This will be, let me just redraw this. Okay, this will be phosphatidylinositol, and this is how a cholesterol bar plot will look like. Okay, let me explain this to you in a bit. So this is cholesterol, this is phosphatidylinositol, this is ethanolamine, this is phosphatidylserine, this would be sphingomyelin, and this is phosphatidylcholine. So here in this graph, you can clearly see that each each phospholipid, no matter if it's sphingolipid, it is a phospholipid, or it is a sterol, no matter what kind of lipid it is actually present in both of the monolayers. But the percentage differs depending on which type of lipid it is. For example, here you can see the sphingomyelin, it is present in higher concentration in exoplasmic monolayer, so is the phosphatidylcholine, but phosphatidylserine and ethanolamine, they are present more on the cytoplasmic monolayer. Here you can see exactly see the difference between the distribution of those lipids. Phosphatidylinositol, this is usually present almost in equal distribution in both of the monolayers, but it shows slight difference, like the gap that exists between the other lipids, the presence of the number of molecules of the other lipids that are present in both of the monolayers, that is quite different. They are quite different, right? So like let's say that phosphatidylserine, out of the 100% of phosphatidylserine that is found in our plasma membrane, 20% is found on the exoplasmic layer and 80% is found on the cytoplasmic layer. So unlike phosphatidylserine, phosphatidylinositol concentrations would be, let's say, 55%, 45% exoplasmic and 55% cytoplasmic. So the gap is quite low, unlike other lipid molecules. But if someone asks this, phosphatidylinositol is higher in the concentration on the cytoplasmic monolayer. Okay, but the cholesterol, which is a sterol, it is present it is equally distributed in both of the monolayers, exoplasmic as well as cytoplasmic. So this is only lipid molecule which has equal distribution in both of the monolayers. So the lipids that have higher concentration in the cytoplasmic monolayers are phosphatidylserine, phosphatidylethanolamine, and phosphatidylinositol. The lipids that have higher concentration in the exoplasmic monolayers are sphingomyelin, phosphatidylcholine. Okay, while unlike all of those, cholesterol have equal distribution in both of the monolayers. Okay, so why this asymmetry is important? Okay, so let's learn about the functional asymmetry, the functional importance of asymmetry in the monolayer. So everything happens for a reason, right? In the cell, if there is some structure, it is important due to in some point of view. So there is some importance of asymmetry. So that is what we are going to learn now. Functional importance of asymmetry. So why not you distribute the lipid molecules symmetrically? Why doesn't the cell distribute them equally? Because it has a very important role in downstream signaling. Whenever a signal arrives at the cell, that passing of the extracellular signal to the intracellular molecules, or whenever any kind of intracellular molecules get activated and it has to signal downstream, in that case the asymmetry helps. So let's now list down certain examples of how in different scenarios this asymmetry is going to play a role. So let's first, for that matter, let's first learn about the functioning of molecule kinase known as protein kinase C. Okay, so as you can see, say that this enzyme is a kinase, means it is going to phosphorylate certain protein. It is a protein kinase, means it is going to phosphorylate certain protein. If you don't know about the signaling pathways of PKC, don't worry. When we'll cover the cellular signaling part, we'll discuss about this in detail. But if even if you don't know the proper signaling pathway for PKC, even if you have a little bit knowledge of what kind of signaling pathways are there, or how the signaling happens, then you'll be able to understand this point. If you don't get it, don't worry, we'll discuss this in cellular signaling part as well. So protein kinase C, so it is a very important molecule. So what happens is that when it is activated, when there is certain extracellular signal and it gets activated, so this goes and binds to phosphatidylserine, which is, and just now you saw that phosphatidylserine is present on the higher concentration in cytoplasmic monolayers. So it goes and binds to the phosphatidylserine molecule, and then it can do the downstream signaling. So for certain, for certain signal to pass to, for so let's say, so this is a protein kinase, right? So it is going to phosphorylate certain protein. So for having the downstream effects of this protein kinase, so what, let's say that it is going to phosphorylate protein A. So for having the protein A phosphorylation, first you need two conditions to, to check box two conditions. First, you need to activate protein kinase C, and then wherever it is getting activated, it there needs the higher concentration of phosphatidylserine on the cytoplasmic monomer. When those two conditions are met, only then you will see the downstream effects of protein kinase C. So when protein kinase C is activated, it goes and it binds to the phosphatidylserine, and then you can see the downstream effects of this protein. So this is how the asymmetry plays a quite important role in the case of cells. Okay, let's now learn about a different example. So there's a so this was a protein kinase. There is a lipid kinase known as phosphoinositide 3 kinase, PI3 kinase. Okay, so this PI3 kinase, this is phosphoinositide kinase. Okay, so you can just by the name you can guess which kind of substrate it is going to phosphorylate, right? So it is a lipid kinase, and that is why it phosphorylates a lipid. The lipid which is called as phosphatidylinositol. So just now you learned that phosphatidylinositol is also present on in higher concentrations on the cytosolic monomer, right? So this PI3 kinase, it goes and phosphorylates PI, the phosphatidylinositol. Okay. So when it phosphorylates this inositol ring, the inositol ring that is present on this lipid, it actually creates a site, a binding site. So this is PI, which is phosphorylated. So this phosphorylation of inositol ring of phosphatidylinositol, this creates a binding site for certain proteins, for certain intracellular proteins. So that the proteins go bind to this phosphorylated phosphatidylinositol, and now they can do the downstream signaling. Usually, those type of proteins, they guide membrane transport, the transport of the proteins. Okay, so they are important. So for getting them activated, they need to go and bind to the phosphorylated inositol. So this is how the asymmetry also works. So what happens is that it is going to create a binding site, which is important for downstream signaling in certain cells. So this is only possible because PI3 kinase, the phosphatidylinositol, this is present in higher concentration in the cytosolic monolayer, not in the exoplasmic monolayer. So this kind of ensures that whatever signaling is going on, it goes inside the cell and not outside the cell. So whatever activating molecules are there intracellularly present, they go and they can bind to the either the cytosolic monolayer lipid, or they can go and cleave that lipid. For example, the phospholipase C. Okay, so you can see it is a lipase, means it is going to cleave the lipid molecule. So this phospholipase C, in this case, you are going to create a binding site, but this is a different case. In this case, it actually cleaves the inositol phospholipids. So this is cleaving. This is going to cleave the phosphatidylinositol, and when it cleaves this lipid molecule, it generates two fragments. So like this is the lipid, this is lipid bilayer. I'm just drawing a single one. Okay, so this is a lipid bilayer, right? So when phospholipase C works on the cytosolic part, so this is the cytosolic part, right? So it is going to cleave the molecule and it will generate two fragments. So it will generate this, okay, and it will generate the second fragment, which will go intracellularly. So it is going to generate a fragment out of which two fragments out of which one fragment will remain in the membrane, right? So it is going to create two fragments where one will stay in the membrane and one will be free now in the cytosol, which is going to carry out the downstream signaling, right? So this is how the function. This is how the asymmetry is quite important for functioning of the cells. So this molecule, which which is free in the cytosol, this is going to now activate the protein kinase C, and this protein kinase C, when gets activated, we saw that when it gets activated, it goes and binds to phosphatidylserine and then does the downstream signaling. So this is how the whole signaling cascade, which is a very complex process which is a very complex process. It is possible to, it is possible and it is very coordinated because there's asymmetry in the membrane. So that is why this is how the uh, plasma membrane is able to differentiate between the outside and the inside. So there should be certain marks, right, on the plasma membrane. So when I'm drawing this plasma membrane, right, let me just give you an example. When I am drawing this plasma membrane, something like this. Okay, and I just draw it like this. So when I'm not drawing a full circle, when I'm not drawing a full cell, you cannot really gaze that which side is extracellular matrix or cytosol. There could be two scenarios. First, that this is extracellular matrix and this is cytosol. And the second scenario is that this could be cytosol and this is extracellular matrix. So for the purpose of differentiate between the different environments, whether this is the intracellular environment or whether this is extracellular environment, whether this is the exoplasmic part of the plasma membrane or whether this is the cytosolic monolayer of the plasma membrane, to be able to differentiate this, you need certain asymmetry. You need certain set asymmetry, which is going to indicate, okay, that if the sphingomyelin is higher on this side, means this is the extracellular part. If the phosphatidylserine is higher on this side, means this is the cytosolic part, right? So that is why the asymmetry is very important, because it sets polarity. And apart from setting polarity in the cell, it also helps the cell to maintain its downstream signaling. So this makes the cell, because if the cell is able to recognize the intracellular part and extracellular part, it is going to differentiate between those two parts clearly. Then it will be able to perform the signaling in a coordinated manner. That is how the asymmetry in the plasma membrane is very important. This is also a very important factor when it comes to in differentiating between live cell and dead cell. So what happens is that when cells are live, they do not show they usually have the phosphatidylserine higher in the cytoplasmic monolayer, right? But when a cell undergoes apoptosis, so this could be the fourth point, right? So when the cell undergoes apoptosis, the phosphatidylserine, which was higher on the cytoplasmic monolayer, it actually flips and it is exposed on the outer surface, because it is going to flip and it is going to reach to the extracellular, the non-cytosolic or the exoplasmic monolayer. So this is exposed on the surface and it provides the kill me signals to the macrophages, because this is not normal. This is an abnormal scenario, that phosphatidylserine should in normally in the live cell it should be higher inside on the cytosolic monolayer, but it is exposed on the surface. This gives the signal to macrophage that this cell is not normal now. So this is the cell which is undergoing the apoptosis, and that is how the macrophages goes. They go and do the phagocytosis of such cells. So when the phosphatidylserine it is exposed on the surface, macrophages get the kill me signal. Macrophages, they have the kill me signal. They will go phagocytose the the cell, and that is how the cell now won't be, it will undergo apoptosis, and it won't be able to evoke the inflammation, because if the DNA or the intracellular contents are spilled in the extracellular matrix, it is going to it is going to elicit the immune responses. But due to the presence of phosphatidylserine on the exoplasmic monolayer, it sends the macrophages the signal that this cell is not normal. Maybe it is undergoing apoptosis, because it is clearly giving you the signal that kill me or eat me. These are also known as eat me signals. So now macrophage will go phagocytose that cell, and this is how the cell is prevented from causing inflammation in the extracellular matrix. So this is how the asymmetry of plasma membrane plays a quite important role. And with this, we are done with the plasma membrane part, specifically the lipid part. In the next lecture, we will start learning about membrane proteins, the different types of membrane proteins that are present in the lipid bilayer. So when we started this series in the cell biology series, we first learned about cytoskeleton, then we started about plasma membrane, right? So in the plasma membrane, we saw that it is composed of lipids, proteins, and carbohydrates. Even though the percentage of carbohydrate is very less, but it is an important component of the lipid bilayer. So the lipids form the lipid bilayer. So they are like the sea in which the proteins are embedded. So in next class, we are going to learn about those proteins which are also referred as the membrane proteins, because there are proteins in the cytoplasm as well. Those are the cytosolic proteins, but these are embedded on the membrane, and that is why they are known as membrane proteins. But there are different types of membrane proteins depending on whether they are fully submerged in the membrane, whether they are integrated with that membrane, means the integral proteins, or whether they are just floating in certain monolayer, means they are peripheral proteins. So we are going to learn about those different types of the proteins in next lecture, and what do they do, like what are their functions in detail. So I conclude this lecture. Till then, see you. Bye bye.